I'm Laura Baggy at Duke University.
I'm Kevin Koset. I'm an assistant professor at the University of Alabama. So it happened pretty quickly. We were descending before I knew it.
Yes, we did not spend very much time on the surface at all. We basically were dropped into the water and we started immediately descending. I think we started descending at a quicker rate than before we got to 250 meters.
So I guess the lights were still off at this point of the dive. And part of the problem is we hadn't quite figured out the camera systems yet. So sometimes, at least speaking for myself, my cinematography is not going to be the best. Okay, now I have lights.
Yes, this is at 200. Is this at 250 meters?
Must
yes.
be, yeah.
We turned on some of the lights at 200 meters of depth.
Okay.
And this is when we're just looking around to see what plankton are there in the water column.
Cool. Yep. And so I was port. I'm on the left. Laura was starboard. You're on the right. Does that sound correct? Oh, is
That's
that a phronema?
right.
Yep.
Yes, and we immediately were able to see the species that I'm looking for, which is phronema, and it's a little crustacean that lives inside of a salp, and you can see it on the port camera there, sort of zooming around. A salp is a type. Well, it looks kind of like a jellyfish. It's a gelatinous animal that the phronema eats from the inside out, and then makes her home inside of there.
I think Jefferson is getting the slurper ready to go.
Oh,
and now he's got the scale bar out. So that was for Joanna Kinsley's work. She's looking at particle sinking rates in the ocean. And so we have this scale bar with regularly marked lines so she can see the particle size and how quickly they're sinking.
Yeah, so we measured particle sinking rate for a while. Before then we put that back, and then we got out the slurp gun again to try to catch some of the high-period amphipods. Hopefully it will come up on the frame grabs. I'm not sure if it will be able to actually see the moment of capture. We'll definitely see that in the whole video. Oh, there it is! There on the port side. It's really exciting. You saw it there for a moment. It was swimming around, and now it's coming up, and you actually get to see it be slurped in.
You can tell I was frantically moving the camera around trying to get it into focus. And I think this must be the point where I stopped playing with the camera because I was taking notes.
Yeah.
So you could see my really nice view of the bottom of the sub.
Oh yes, okay, so what's happening here when the lights go back off is I was making observations of bioluminescence, and this camera is not strong enough to actually pick it up, so you can't see just how amazing it was, but we were seeing what the bioluminescence response would be to flashing the Alvin lights, and you could see when you turn the lights off and turn them back on, the animals just lit up like crazy. Oh, there's a phronema again! So this is when we caught our second phronema, and that passed by really quickly. But we were able to successfully collect 2 phronema specimens at that depth. So that was really exciting. The density of animals in the water column was just amazing. All those little bits you see floating by, a lot of them are crustaceans. The phronema was the easiest one to see because it is in that gelatinous salt barrel, but all the other free-swimming hyperids are just everywhere. So inside Alvin, I thought it was going to be claustrophobic at first, but as soon as we got in the water column and were able to look out the window, it just seemed like we were in this giant world of outer space, actually, especially when we were looking at the bioluminescence, because it didn't seem cramped at all. There wasn't a lot of space to move around, but it was pretty comfortable, and as we descended and we spent about 3 hours working in the midwater. So from the surface to the time that we got to the bottom, we spent, I think it was 3.5 hours descending. At this point, we are descending with the lights on at a rate of 15 meters per minute, and we are making observations of what kind of plankton are there in the water column.
One of the other observers has commented that people often think of the ocean as being this, sparse wasteland where there's not a lot of life, suspended in the water, but it was packed with plankton. We were just amazed at how much life we saw.
Yeah, it was very dense, and you could see sort of distinct layers of different types of organisms.
So at this step we saw some squid.
Oh yeah, some juvenile squid.
And I believe an expert identified this as Goniatus, is that right?
It's a buried toothus squid. And then there's another dense layer of crustaceans that we passed through. Yeah, descending through the dense layer of crustaceans was very exciting for me. I guess it wouldn't be like a hailstorm because it wasn't hitting the sub or anything. They were swimming out of the way. But it's kind of like going through a snowstorm, I guess, of crustaceans.
Yeah, that's a better way to put it.
Yeah. The little flicks that you see on the screen are Hyperiid amphipods or some other kinds of midwater shrimp. So here we're approaching 500 meters, which was our second stop point on this dive. We had chosen 3 distinct depths where we wanted to stop and spend extra time sampling the water column. So those were 250, 500, and 750 meters of depth. And at those points, we looked at the scale to measure particle sinking rates. We sampled the plankton there, trying to get specifically my Hyperiid amphipods. And we also observed bioluminescent response, and we noticed that at a 500 and 750 meters depth, the bioluminescence was just even greater than it was at 250. It was just amazing. You could see the distinct outlines of all the animals. Yeah, and we noticed once we got past 550 meters, a lot of the crustaceans went from being transparent to being a deep red color, which is very interesting. So that's a strategy to avoid being reflective to bioluminescent flashes. And you can see that on the screen right now is just all the red crustaceans swimming around. The red is absorptive, so a lot of the bioluminescent flashes are blue-green, so rather than they don't reflect that light back to the viewer.
One cool thing we saw at 750 meters was pipefish, which I thought were only in shallow water. But evidently there's deep-sea pipefish, which are seahorse relatives.
One of the coolest things we saw was at the end of the dive, but I think it was around this depth, was one pipefish eating another pipefish. It looked pretty scary.
I was worried about being claustrophobic, especially the first time I got in the sub and saw it's actually a lot smaller on the inside than you would guess from the outside. But no, it was just a really cool experience. I think we just were distracted by everything outside to worry about what was really going on inside the sub. Port side, it's looking to the right, looking towards starboard, and there's shrimp attracted to a light there.
Yeah. And I think, what did we catch at 750 meters? We caught a lot more of the Hyperiid amphipods at that depth as well. And we also caught a siphonophore that unfortunately broke apart in the slurp sampler. And you just see how dense the crustaceans are, especially. I mean, there are a lot of things in the water column, gelatinous things as well, but most notable, I think, were the crustaceans. Would you say? Yeah.
I agree. On the right, top right, is the track of the sub. They keep track of the coordinates of the sub in terms of X, Y, and so that's meters from an arbitrarily selected starting position. And so you can see the track that we've moved along as we descend. So we're just seeing X, Y, not Z. Of course, if we're seeing Z, you'd see us going down as well. This is more useful when you're along the bottom, trying to figure out relative coordinates from where you start the dive. Usually the dives begin quote-unquote on the bottom, but we were doing something unusual and doing this midwater work as well. So our dive really began collecting data as soon as we were in the water. And we took turns like stretching our legs out and standing up a little bit. Our pilot Jefferson must be really used to this because he didn't move that much at all, really. He stayed in his pilot seat, occasionally leaning over, looking at the port and starboard portholes, but he's clearly It's not his first rodeo.
Yeah.
So we're getting close to the bottom now in the video.
Yeah.
I believe our bottom depth is approximately 980 meters.
Yeah, I think it topped out at 950. That was our target depth, but we had moved a little bit while descending, and so we landed closer to target 2 than to target 1. When we landed, we did not bump the bottom, so Jefferson was very skilled, our pilot Jefferson was very skilled at sort of making us neutral before we got to that point. But we were sighting the bottom, so we were both had our faces pressed against the glass almost, trying to look out and see when the bottom was approaching.
One of the objectives of our dive was to collect some push cores, these T-shaped handles on tubes. That was a lower objective, like a lower priority objective on our dive, but we landed in an area with a bit of sediment. So we did that pretty much right off the bat. And here we are now collecting the skeleton of a hard coral that had fallen into the sediment. There's a member of Team Benthos who is studying corals using their skeleton to age them. And so she's going to do radiocarbon dating on that specimen, which you can see we've put into the bio box here.
We saw a lot of the Atlantic red crabs crawling around, and one of our objectives on the dive was to try to collect them, but they're very fast and don't really want to be collected. And so anytime we try to go out with the manipulator arm to pick one up, they were able to run away before we were able to actually successfully grab any one of them.
Around 1556, we observed some fish taking bites out of the sediment, which is something of interest to another member of Team Water, Amanda, who's studying fish behavior and the interaction of pelagic and benthic animals.
So at this point in the dive, I think we were transitioning to our next target site. Is that correct?
Yep.
To look for the corals.
Yeah, so back in 2013, there was a dive by the Okeanos with this a remotely operated vehicle at this exact spot, this rock formation, where they observed some corals with a particular type of mollusk on them. These are worm-shaped mollusks called Aplacophora. So that Latin name means without a shell. And these are really weird mollusks that I work on. And they were here in 2013, and I was hoping they might still be here, but not even the corals they were on were still here, so they've moved on.
I think we moved, at this point in the dive, we moved away from that site, and then is this when we went to the rock wall? Oh, there's an octopus. So one cool thing that we saw were several different octopus species, but they weren't the finned octopus that Stephanie was looking for.
Yeah, so here we are approaching the rock wall, which was really cool. You can tell, okay, here we go. So there's some really beautiful glass sponges here. A colleague of mine works on these. I hope you can tell us what they are. It was just covered with corals and sponges and all sorts of other animals, lots of shrimp. And so we get to work right away collecting some various corals. So here it is. Here, Jefferson's opening up the bio box to put them into. Oh, we did push core first. That's right. Sea urchins, sea stars.
Yeah, these rock walls were very impressive. They were very tall.
I really have no idea how big they are, but pretty large. We couldn't really see where they lead because it slopes up and away.
Yeah, slopes away, so...
It's a canyon, so I guess it's just kind of the base of the canyon, really.
Mm-hmm. There were a lot of juvenile squid that were living around the corals on this rock wall, and you could just see them swimming by. I kept filming them.
You can tell I spent a lot of time looking out the portholes at corals and not as much time paying close attention to my camera here. There are a lot of sponges and tunicates on the rock wall as well.
And I spent a lot of time trying to film the shrimp that were associated with those corals and sponges. You'd see little groups of 3. A lot of funny looking lobster as well.
We tried to sample a few hard corals, but they're a bit challenging to get because they're brittle and break easily. So I think there you see some coral that we tried to break off the rock wall, but unfortunately it shattered. But Jefferson was able to pick it up and put most of it back into the bio box.
One challenge of working in Alvin is that sometimes if you stay in one area for too long, you can kick up the dust on the bottom, and then that's what you're seeing in these photos here. It makes it a little bit hard to see what you're doing, so you have to move on to a new spot or wait for the dust to settle.
The lasers you occasionally see in the photos are attached to one of the cameras, and those are used as a scale. So those 2 lasers are 10 centimeters apart, so you can get an idea of the scale of some of the things we're seeing. The pink mushroom-shaped corals are called anthomastus, so they're a type of soft coral. The other, also kind of mushroom-shaped white corals are called cup corals, and they have a hard skeleton. They're a hard coral. There are a lot of really nice bubblegum corals, or another type of soft coral. And this orangish thing down there with that looks like little feather-shaped branches coming off of it, it's actually a sea star. I thought it was a coral from a distance, but it turned out to be a type of predatory sea star.
You can see in the starboard camera that I am videotaping all the shrimp which are associated with those corals, usually in groups of 3. The grouping of 3, I'm not sure what it means. I just thought it was interesting that that's what I often saw them gathered in a little group. Sometimes shrimp gather together, sometimes they're solitary. I don't think we know very much about this particular shrimp species and what they're doing. Inside the sub at the bottom, it finally got a little bit colder at this point. When your arms were near the porthole windows, at least, it got quite a bit chillier than it was descending through the water column, so I think both of us put on sweatshirts and other warm clothes at that point.
The walls of the sub itself were pretty cold, so a lot of condensation started forming. Our pilot Jefferson pointed out to us early on in the dive not to be alarmed if you see water around the portholes. That's not leaking, it's condensation forming. I'm glad he pointed that out though, because I might have been a little nervous otherwise.
Exactly.
So this is another really good spot. You can just see that the rock wall is covered with different kinds of corals and all sorts of invertebrates, so it was really spectacular to see. We were hunting after a few particular kinds of corals, in particular black corals, which are a group of very slowly growing deep sea corals. And unfortunately, it's really hard to tell from the portholes of the sub, even with the HD camera, what you're seeing. With some of these corals, you have to count the number of tentacles they have and things like that. So we tried really hard to do our best to identify things, but we actually collected one coral that didn't end up being what I thought it was, unfortunately.
Sometimes it was hard to tell the color of the animals from the sub.
Some things that looked white in the water when we reached the surface were very pink, which really surprised me. So one thing you can't really see, but we were also occasionally doing what's called tripping Niskin bottles. So we collected water samples throughout the dive.
I think this is when we're moving to one of the sites to collect more water.
Yeah, that's
right.
Before
we go back to the surface.
We just lifted off the bottom a little bit.
Mm-hmm. Yeah, because we went to one final site.
Right.
Near this point in the dive, we were able to catch a really cool baby lobster. or a lobster larva. And we couldn't really tell what it was swimming around because it was zooming around so fast in the water column. We thought it might be a high-period amphipod, but it turned out to be just a little baby lobster. We weren't specifically searching for this baby lobster. We collected it because I thought it might actually be a high-period amphipod, because I could tell it was a crustacean and it was swimming in the way that high-period amphipods normally swim. But it turns out that I have a colleague who's actually studying the specific larval stage of lobster, and so the animal that I collected will be of use to this other person's scientific study.
One cool thing we noticed on the rock walls was some vertical zonation of the corals. So some corals are only lower, some corals are only higher, some of them are only under overhangs, it seems. So they seem to be pretty specific about where they grow. The yellow coral that we just saw quite a few of, one of those, once we got it back to the lab, turned out to have one of these weird aplicophoran mollusks, the worm-shaped mollusks I mentioned, so that was exciting for me. Aplicophoran mollusks pretty much only live on corals. There's some that burrow in the sediment, but the group I work on pretty much only live on corals, and they feed on them.
The machine on the right is the slurp sampler, and on this dive, I forgot to mention earlier that this slurp sampler had 5 different chambers in it, so we were able to switch around and collect animals from specific depths and know which animal was there. So typically on dives, Alvin only brings down the 1-chamber slurp sampler, and so everything that you slurp up with this little vacuum cleaner gun goes into 1 chamber. But here we were able to switch out and know that we caught only 1 or 2 things at specific depths. Okay, so at this point in the dive, we were at the very end and we were just about to ascend. We were doing a final push core, and something interesting happened. The basket, or the tray sort of carrying all the scientific equipment, broke. It just snapped, and we don't really know what happened, but you can see it's kind of hanging off here, and so our pilot Jefferson had to grab the basket with both of Alvin's manipulator arms in order to hold on to it so that we could bring it back to the surface and recover it.
It was a bit unfortunate that it happened at that time because the bio box hadn't yet been closed, and so our samples are sitting there open and exposed, and we're worried that once we reached the surface, the waves would knock them out of the box. And we actually did lose 1 coral specimen, but otherwise everything stayed in there, which was great.
It took about a half hour to ascend to the surface from our final depth, which was at about 850 meters, I think, was the point where we started to ascend. So it took, about a half hour.
Because of the problem with the bio box, we ended up sitting on the surface for quite a bit, and so I was trying to emotionally prepare myself for that because I get seasick, and Alvin tumbles around a bit on the surface. Not tumbles, but certainly bounces around a bit.
It rolls. It felt like a roller coaster ride at State Fair.
We were bouncing around for a while, and I totally got seasick. I didn't throw up or anything, but I was getting there. I was glad we got out when we did. Being lifted out of the water by the A-frame totally reminded me of one of those rides you would go to at like Disney or some amusement park where you're in like a closed box that's on hydraulics, like twisting and turning and things like that. The sub was kind of lurching at different angles and things, but it was fun. It was really cool. It didn't take long for us to get out of the water once we were hooked up to the A-frame, and
because we had been on the surface for a while, rolling around while the swimmers were securing the basket and securing all our boxes of samples, it felt like a relief to be lifted out of the water on the A-frame. It felt kind of like a ride at a state fair being over. But we were very sad to leave the water. I know I wanted to spend more time in the sub.
It went by really fast.
The whole process of being lifted out of the water and put back onto the tracks was very gentle. We didn't really notice anything at all inside the sub.
Aside from a little rocking, yeah, it was very gentle.
and I'm like, you guys want to go together?
Yeah, I think, you know, Team Inverts needs to get wet together. It just keeps coming. Oh, more, really? Oh, come on.